fact that we sequence the human genome. And we thought we would find all the answers to all our questions about human health and disease, but we didn't. We don't know everything. We don't know what lies in our genome, what makes us what we are. What are the hidden messages in the genome? If you take an adult organism, and if you look at all the different cell types that we have, they have all the same, the same genes, all of them, but they look different, they function differently, and they know what they have to do. So what makes them different? What makes them look so different and make so different types of organs? What is their genetic program? The genetic program starts in a moment of fertilization when the sperm invades the oocyte and the two genomes will unite. And the genetic program will create a series of events that will lead to the formation of the whole organism with all the different cell types with the shape that we know. And we have 200 different cell types. But all of the cells use the 20,000 genes. They need all the 20,000 genes in order to function, but they will select. So one cell does not use all the genes, but will remember which ones to use. For example, the kidney cell will use the gene B, but will inactivate the gene A and C. The liver cell will use only gene C. These are just examples. And the brain cell will use only gene A and C. In case this memory is lost, in case the cells don't know anymore which genes to use and which genes not to use, a situation like cancer will arise. For example, in this case, everything is the same except this gene, which is inactivated in a normal cell. But in case it is activated, there will be an altered genetic program that will lead to a cancer situation. And a lost memory means a lost identity. I don't know anymore what I am. I become antisocial. I will overgrow the normal tissue and I will become a tumor. So what is actually the memory of the cell? We know how it looks like, but molecularly, what is it? So scientists have been looking at this, and epigenetics became quite intensely studied in the last 20, 30 years. So if you have our genes, they have certain signals called the promoters that will tell, like you have a train, you have an engine of a train, an engine will tell what the gene has to do. This engine can have a traffic light system. So basically these signals are so-called methyl groups. There are some chemical signals that are attached to these promoter regions. And then they will tell whether the gene should be active or inactive. In addition, there are some proteins called transcription factors that will bind to these promoters according to the signal that they see. So if they see a methyl group, for example, then there will be a different transcription factor binding it. In the moment when they bind, the expression of this gene will not function anymore, whereas the green ones will function. So this is how we make the difference between different cell types. So basically the molecular traffic system forms a gene memory, which is above the genetics, therefore it's epigenetics. In addition, epigenetic memory is quite persistent. That means that when the cell divides, it has a memory in the original cell, but will transmit this memory to the next cell, to the daughter cells. As you see here, this is a dividing cell, and these are the chromosomes. You see these like yellow signals. They are the traffic lights that hang onto the DNA when the cell divides. I will be just talking about DNA methylation, but there are some additional known signals so far, and a system is much more complex, but I will just stick to the DNA methylation. So I will just summarize some of the phenomena that exist related to the epigenetics, and they are of medical importance. And in addition, probably it's very good for us to know what can happen in addition to the gene function. So as you know, we have chromosomes. We inherit one from the mother, one from the father. And as well, the genes are the same between the mother and the father, and we inherit from both of them. There are some certain situations where some genes function differently, whether they come from the mother or the father. Like in this case, this gene is activated, whereas the one that comes from the father is inactivated. And this is important to be maintained. And this arises very early in our life. And if that doesn't happen, then certain conditions like the Becker-Williams syndrome can form. So this gene got activated, and this baby has these malformations. What is the reason for this genomic imprinting? The most accepted theories up to now is a so-called parental genomic conflict. That means that the genes that come from the father, the genes on the father's chromosome, the ones that are active, they will want the baby to be big, they will want to take more resources from the mother. Whereas the genes from the mother, they want to preserve the mother, to have the right size, so the baby has the right size so that it doesn't take so many resources for the mother, so that the mother can survive as well. Right. We have seen monozygotic twins that they look alike, but very often that's not the case. And then we think, what is the reason behind? They have the same genes, they're identical. It happens often that one is healthy, one suffers of diabetes, one is depressed, the other is not. Like in this case as well, there are also discordant people with Becker-Williams syndrome. One has the illness, the other one doesn't. And it's an imprinting that arises in the womb very early in development. As well, it is quite known that culturing cells in the dish might influence the epigenome. So if you know about the term genome, which is the genes, there is a term epigenome. There is a coding, a traffic light system. It's very complex about the genome, and that also might be influenced by the environment. This is not a great concern, but it's something to keep in mind. There is a higher incidence of some diseases in terms, when it comes to IVF. As well, aging. In aging, people have seen researchers, they have seen modifications that arise at the epigenome. In case of the common illnesses, schizophrenia, diabetes, cancer, depression, there are some epigenetic factors involved there. And this is a great example with an agouti mice, where this is the agouti mouse, it's a yellow mouse that is an experimental model for obesity and diabetes research. They are prone to develop cancer. In an experiment, some scientists just feed the mother with dietary supplements. And these dietary supplements contain the so-called methyl donors. And they realized that if they added that to the mother while she was pregnant with babies, the pups were healthy, brown, and thin. In case they didn't add those supplements, they were just like the mother. So it's something to keep in mind that environment can influence quite a lot. As well, making of a dolly is based on an epigenetic phenomenon. And the way they did it, they took a mammary gland cell and they put it into an oocyte that was enucleated, so the nucleus was taken out. And the oocyte has the capability of reprogramming the cell, meaning that the oocyte, if a foreign nucleus is put into it, a somatic, from a developed cell, from a differentiated cell, it will erase the memory. The process is very inefficient, but it does happen. And that's how Dolly was born. It took around 400 conceptions until living animal survived. Right, stem cells, they are well known, probably, by you as well. And they are of great medical importance nowadays. So I just wanted to show you where they come from and why are they so important. So when the sperm fertilizes the oocyte, there is a so-called zygote stage. That's the only stage where we have totipotency. That means that that cell can generate any type of our body plus placenta as well. The more the cells divide, the more they lose this totipotency, and then soon will fall into the pluripotent state, and that's the blastocyst. The blastocyst contains an inner cell mass, which are basically the stem cells. So if you take out the inner cell mass, you can culture it in vitro. This is a real blastocyst, and this is the inner cell mass. So from the blastocyst, the adult organism is formed, and we do have adult stem cells as well. Adult stem cells will give rise to differentiated cells when they are damaged, when they are apoptotic, when they decide they don't want to live and replenish the body with new cells. That's how we regenerate. What scientists have found, and that's in the last 2-3 years, that if we take adult cells, and not like making dolly, but by adding a cocktail of proteins, we can de-differentiate, we can reprogram cells to the level of pluripotency. So normally the sources of stem cells are adult stem cells, embryonic stem cells. Adult stem cells are hard to get into culture, and embryonic stem cells are prone to ethical dilemmas. Umbilical cord blood stem cells, one has to extract it when the baby is born. But it's a very useful source of stem cells, multipotent. Amniotic fluid derived stem cells, one can damage the fetus with that. Somatic cell nuclear transfer, that's how dolly was made, but one needs a lot of oocytes for that, so it's not very practical. And the iPS cells are the so-called induced pluripotent stem cells, and they arise from differentiated cells by reprogramming. So basically, these iPS cells are now a big thing in the biomedical field. They avoid the immune rejection, and they avoid as well ethical problems. Basically, a Japanese group, they thought, okay, we have a stem cell and it works. So what are basically the main genes that can contribute to this pluripotency? And they have selected 24 genes, just according to their thoughts. They have selected 24 genes. And one by one, they went down, and they found that actually 4 genes are enough by putting it on an adult cell to reprogram the cells. And that's how they got induced pluripotent stem cells. Of course, the clinical applications are very promising: generation of new tissues and organs, cell replacement therapy and drug discovery, and most actual use where it is for drug screening. So iPS cells, they have passed the most difficult test for pluripotency, and that is very recent. That actually happened a few months ago. They can be generated from the patient's cells, and as well they can differentiate it to different types of cells. And this is a great source of cell where one can analyze the disease of the patient in a cell culture. It can be used for drug therapies to choose the right drug therapy for that particular person. And it's a potential for tissue support, cell replacement, and regeneration. We have to keep in mind, though, that we are very early in this area, and although we can do quite a lot with stem cells, we still don't know enough about it in order to implement it for safe therapies. We have to know more about what is their identity, what makes them stick to that identity, and it's not an easy task for scientists. And I guess that's where I finish my talk. And this is my lab, this is the institute, and I want to thank you.